the progress that can be made if the molecular techniques come together, and that you can see that classical oil palm breeding is one thing, but what can be made now? But I'm afraid for the discussions there will be some questions. Oh, I'm sorry. I thought I was unmuted by. Okay, thank you very, very much. I'm afraid you didn't hear me when I was talking on the progress that the molecular science coming together with the breeding and all the efforts that farmers there had done till now, that the progress gives a lot of hope. So for the discussions, I think the panelists can start, but of course, I take the privilege of being the first one, asking, it's good to see how you can work now with the elite germplasm you have and how you can make better use of it. But would it not be also worthwhile that you would join the action that some molecular work would be done to create new varieties with all the elements that you have now found, that by saying transgenics and make it in a way that you keep a maximum the elite variety, add some traits, because 25 years is still a lot of work, a lot of time before you harvest it. And for environment, your work shows how urgently it is for the whole area of Malaysia and Indonesia and other parts of the world to have high yield, so that you really use less land and that this land can go back to wild forest for biodiversity, all the problems that some scientists have identified now, and that we want environmental changes for it. So doing that, I think it's also urgent that the research on adding or altering sequences also can go on. Who wants to take that up?
I could say a couple of things right away. I mean, you are, of course, exactly right. I mean, we've identified, just by looking at natural variation, very, very similar to Sarah's work with maize, lots of opportunities for genome editing that would allow us to immediately introduce beneficial traits into elite germplasm. But at the moment, genome editing is still not, it's still classified as GMO. And as long as that's true, export crops such as palm oil, or export products such as palm oil, are going to have a hard time being able to use this sort of technology. I think it's really important to keep lobbying, especially the Europeans, to allow or change the regulations on genome editing. But as you could see from our talks, I mean, there are so many opportunities, and it will greatly accelerate most of the breeding schemes we have. So I think it raised a really, really important point.
Yes, but I'm afraid that lobbying is not enough, because that's 30 years of lobbying that was done by just argumenting with scientific arguments. And the answer we receive show us that the product is there, and then we will judge, like we do for all the products that material sciences has made through really all the possible scientific approaches that you can do there. So they say, show us, and to show us, we should at least obtain, and I think in Malaysia and Indonesia, it would be perfect that if authorities say, at least we go for this work to make the prototype, okay, it can take 10 years, but at least it has been done. It's now there are so many new things that you will identify through the molecular techniques. You have now two traits that you can follow, but we know the way epigenetics is going, that we will understand better the gene expression in plants. And at that moment, if we do that, we will do a lot of alterations that will make better stocks for starting, better germplasm. And a better germplasm you can make in greenhouses for Arabidopsis, but for oil palm tree, you have to see the real facts. And that should be possible to communicate. So I think it's an enormous task for the younger generations to say, we have to explain what is the science. And if you claim that there is any danger, show us, explain why you see that there would be danger.
If I could just add to what Rob said, and I agree. Well, although commercial palm oil is GM-free, but we have always believed that the science must go on. And MPOB actually has got work. It started many years ago on actually producing transgenic oil palm. But in this case, the target was more for tailoring oil composition. So we have that project, and we have putative and transgenic oil palm in this greenhouse, but not for commercial purposes. And there is also work now on genome editing, also for oil composition, and other traits like resistance to Ganoderma. I think there was a paper by the Indonesians on this. We're not sure if they've demonstrated resistance, but there is work on genome editing for Ganoderma resistance. Ganoderma is the most serious pathogen of the oil palm. So work is going on. Because as you say, the oil palm is 25 years and we really have to start now. So the science is going on. And perhaps by the time the EU and the rest of the world are accepting genome editing, I think MPOB would hopefully have the science all worked out. Yeah. Mm-hmm.
Yeah, but it's indeed, if you just do the science, you already can convince a limited number of the populations, and there you will have persons that find the right words to explain it to society. But it has gone. Well, I don't want to mobilize the talks, please, among the panelists. What ideas come up for you, among yourself that your science is bringing, and that should come up, and that the society should know that any, or should I go on doing questioning?
I can see we do have some questions.
Yeah, I can actually get started with a few questions. I have a few of my own and a few came in from the audience, but let's start with one from audience member Doreen Ware. This is directed to Sarah. She says, beautiful work and talk. Based on your initial screen of the environmental factors using the GEPV, how many breeding cycles are needed to get germplasm that breeders are willing to use in their programs?
Okay, very, very good question, Doreen. So the current number of generations that we've been able to really achieve a product that is semi-elite, that's uniform, that breeders are willing to use is as little as 4 generations. So that, as we can in the field here push easily to three cycles a year is pretty fast. The challenge has been, up until very recently, we haven't been using and leveraging a lot of the genomics in that. We have been doing a lot of field phenotyping, which we can only do once a year. So that limits the time, because we have a certain periodicity that we've got to adhere to. So the products that have come out now have been developed across about, let's have a think, 7 years of research efforts. Now, as we start to move these things more into a parallel structure, and rather than the classic, we will develop a population to really understand what the biology is and to start to dissect that more cleanly at the same time, or earlier than developing populations that breeders will actually use, we're combining these things together. So we're discovering as we are breeding. Nurseries themselves are cheap and easy to make. Yeah, making crosses is not difficult. Making the progeny is not expensive. The challenge is in the phenotyping of that. And the cost benefit really is how quickly can we get a product to breeders. So we're starting to run these things in parallel, phenotyping and validating as we move forwards, using the understanding that we have to select the higher value starting materials and the higher value alleles to focus on as part of that process.
For the corn and wheat problems, there is so much work going on all over the world. I think it also should be very important that you can come up and really explain how the environmental advantages are for using these varieties, and for particularly for the areas like Africa, where they cannot at the moment, through the history of the country, develop the science themselves, but all scientists are in their solidarity could build up together efforts, like in the beginning of the CGIAR ideas were there, but now the tools have progressed so nicely that you can do demonstration plots and in contained conditions that people should be really convinced that the science contributes to the environment, and that in the timing you do it, that it will be much better than making rules of we have to do mitigation for the CO2, and we have to wait till the whole industry reconstructs. We can say that I'm convinced we have, with this and this time scheme, we can get positive results with the science we are doing. Can you convince at the CG system that this attitude should be taken?
I mean, the CG system is science driven. Yeah, we use science as a guide.
I'm convinced.
But we have to, within that context, respect national laws, and we do partner with varying countries in transgenic and gene editing products. We've got a number on the way for both maize and wheat, at least from the CIMMYT perspective. But obviously we do that only where and when a country has requested use and development of that particular approach. That said, across the CG now, we have a number of collaborations with groups like IGI at Berkeley, where we're exploring the use of technologies like gene editing for a variety of approaches. And one of which I mentioned is the validation. We can identify what we think may be a high value trait in a wild relative, in a landrace. That takes a lot of effort to get into elite germplasm. Now, through substitution and other technologies, we can quickly validate the effect of that in an elite background, see if it's really worthwhile. Now, that product can, if there is the appropriate legislative framework, be deployed in partner countries. Where there isn't, at least it gives us information that this particular allele or set of alleles are high value, and we need to start developing that pre-breeding process. So through the use of this technology for validation, we can increase the familiarity of different stakeholders of the technology and let them make their own science-based decisions in terms of what they want to adopt nationally.
Yeah, I'm convinced absolutely that you can do, and I consider that as at the moment in despair, the only thing that can be done. But you could think in the way, if there are unknowns and for all society problems. There are so many announced where in the human sciences that we don't know what is better, this and that. And we also have to make decisions. So explain that the product is there and really take a leading position in demonstrating that you can make the product and challenging them with the arguments that they are considering as most valuable. You cannot explain you want more yield because that is an economic political system. But you could explain for environment, if you want to have these goals, if you could make this, then it will take you only so many years or such time to have that in a large scale. I think that is waiting which organization will take it up and explain it with all respect for why people are afraid and don't understand this type of arguments. That is sure that we have to do that. That's the way our society works. But if you don't go on explaining it and say that how urgent it is and that you see what is the problems with overpopulation, what is the problems of poverty, what will your society if you don't change that see with environment that goes on and use the arguments that could maybe convince the major stakeholders in society.
All right,
Thank you, Mark and Sarah. The next question, this is for Ravi and Rob. So first of all, very interesting talks on the impact of genomics on sustainability. Now, you probably agree that for this approach to succeed, having a complete and well-annotated reference genome is critical. Now, this is possible with technical advances in sequencing techniques, but this could still be a biological problems for complex genomes like, polyploidy or duplications and such. So could you please speak to this challenge and possible ways to overcome this?
Sure. I mean, if you don't mind, Ravi.
Please.
Absolutely. I mean, the oil palm genome was actually done with a combination of next generation sequencing and much more old-fashioned sequencing as well. And these days, for example, polyploid genomes are completely accessible. We've recently in my lab, we've just recently sequenced some polyploid aquatic plants. And you can really phase genomes incredibly well now for very low cost. And I think this really opens up opportunities for agricultural biotechnology in ways that weren't around before. There are also new ways of epigenomic analysis, which also allow very rapid. Again, we use some pretty old technology with the CIMMYT discovery, but the possibility to do that now is much greater. I noticed there was a question from Sebastian Marc as well on how much other plants, other crops could benefit from this sort of approach, and I think it's going to be huge. There's a lot, for example, there's a lot of plants that are micropropagated by tissue culture. The biggest interesting is actually the orchid, which is literally tens of millions of clones of orchids are propagated every year, and this sort of technology would be very useful for orchids. Orchids have huge genomes which were completely inaccessible until very recently, and now they really are. So yes, I think this holds a lot of promise.
So Rob and Ravi, if I can just follow up. Murali Murugavel asks, what is the water use efficiency of oil palms versus other plant oil sources such as soy? And then a second part to Murali's question is, can oil palm plantations actually grow in land-rich locations like Canada or Australia?
First question is for the water use efficiency. well, we haven't really done a comparison between soybean and oil palm, but the fact that the oil palm is a perennial crop and it's got a very deep root system. So I believe that the water use efficiency is actually is better than soybean. It's a perennial crop with a good root system, right? As for the question on whether it can be grown in Canada and other land-rich areas, no. As I explained, it's because the oil palm grows in, I think, best 10 degrees north and south of the equator in the highly ecologically sensitive areas of the world, unless you come up with something that's that can grow at different temperatures. Very, very unlikely that you can grow it in Canada simply because of the temperature, or Australia. I think there is some oil palm in Australia, though, in the warmer areas. But generally, the question is no, I don't think so. Rob?
Yeah, I mean, we have occasionally thought about not just oil palm, but things like sugarcane, changing. We can certainly do a lot with flowering time. We understand that extremely well now, and photoperiod dependent and temperature dependent flowering. But having said that, that's not the only thing. And oil palm especially seems to be very sensitive to a lot of climatic variables that are not fully understood.
Yeah.
So it will be a challenge to engineer some of these crops to grow in other places. It's amazing how maize has been able to
oil palm solidifies at low temperature.
Sorry?
Did
I hear? Couldn't quite hear.
I didn't hear that.
Plus, also another thing is that if you plant it in the cold, because the yield will be low, but we also find that actually the oil palm has got a cold activated lipase. So if you're going to grow it in cold climate, there's going to be a very high free fatty acid content, and that's what produce low quality oil, even if you can produce. So there are many, many things that you have to sort out if you're going to try and grow it in a place like Canada. Even if it can grow at low temperature, there would be an effect on lipase and things like that. So I think the answer is no. Mm-hmm.
Thank you, Ravi and Rob. Sarah, here's one for you. You talked about a nice approach, a data-driven approach, combining native genetic variations with principles of modern informatics to identify elite breeding lines. We heard about this approach with respect to maize. It'll be interesting to see how this approach has been translated to other crops. And if so, are there any challenges?
Good question. So obviously the first challenge in this approach is having collections that have good georeference data. I've recently looked at the wheat collection of the CG, which is well over 100,000 accessions. Only about 6,000 of those at the moment have good collection site information. A lot of this information got lost as it's been translated from the paper records into the digital records. So we're going back and pulling out this valuable information now. So that's the first thing that you need to have. The second thing among those collections that have georeference data is sufficient representation of environmental climes that you're interested in. If most things have come from environments that are particularly uniform, at least across that collection, you're not going to get particularly good return of signal. And then the question is, what is the cline in terms of FSTs across these materials? Maize is particularly good. We get very gradual shifts in allele frequency, so we're able to pick out a little bit more nuance in the analyses. That said, we've been looking at proof of concept of this in other species outside of CIMMYT. We've got good proof of concept in sorghum now, in cowpea, in bean, and we've got a number of projects just getting off the ground now, funded by a number of international donors. So we're expanding the range of crops that we're applying this to about 9 different species now that are some of the core fundamental species for at least human nutrition in the global south.
Thank you.
All right, I have one last question before we conclude this portion of the program and move on to our Meet the Panelists session. excuse me, and that is, can you guys elaborate a little bit on some of the major challenges in translating genomics research, say in oil palm and maize, to commercial outcomes? I'm sure there are many, and we can have a whole discussion on this, but if you could just tell us a little bit about some of these challenges.
Sarah? Maybe I'll have a go then. As far as the oil palm is concerned, obviously DNA-based technology involves high startup cost. And I'll just talk on based on the SureSAVIT series anyway. It involves high startup costs. So that's obviously a major challenge. But it wasn't the most important challenge as far as commercialization of the SureSAVIT series was. And I think one of the problems we have is that the oil palm is a perennial crop. And if you plant it, you don't see the effect till 6 to 8 years down the road. And so there's sometimes a reluctance to do things differently because you're not going to see your results till from 6 to 8 years down the road. So while many oil palm companies are very keen, but you also have people who feel that, well, we're not going to see it while we are the manager, like you say, the managers of these companies, their KPIs are based on what? Short-term performance. They're not going to wait for 8 years to see the difference. So I think there is some reluctance in some parties to implement this technology. So adoption can be a problem because of the perennial nature of the crop. You don't see the results straight away. It takes about 6 to 8 years before you see something. So that's one of the challenges we have as far as translating the genomics work to, let's say, to do a routine DNA testing. Yeah.
And if I take that further, I mean, we see some of the same challenges in the annual crops. I think one additional challenge we have, and in particular with maize, it being such a well-studied species, is there's a lot of the time a disconnect between what are the real needs of the breeding programs and what things are actively being researched on now. So lack of talking to breeders, talking to market specialists, and everybody has their favorite trait. And in some cases that trait is super interesting from a biological point of view, really understanding the mechanistic basis of gene regulation, but it's not something that the breeders need. Flowering time is a great example. We've got millions of studies on flowering time, which is super important. Every breeder I know working on maize says, I can measure that in the field really quickly. I don't need DNA markers for it. So it's trying to identify what are those key points that are really valuable to breeders and are really valuable biologically, and making sure we use those as proof of concept examples to drive technologies forward. Because there's a lot of things that sit on the shelf and get caught up in more hype than anything. So it's really identifying those high value priority cases and moving them forwards effectively.
Yeah, I can only add to that by saying that I think just from the purely technological point of view, agriculture and sustainable agriculture has benefited enormously from developments in human and biomedical research. So sequencing technologies, diagnostic technologies, all of that, and the very high throughput sciences we can bring to bear. So in a way, at least for agriculture, the real rate limiting step is in regulatory and market approval. And that's sort of, it's out of the hands of scientists in many respects. And I think Marc brought out some really, really important issues there that we need more communication between the biotech sector and these regulatory bodies, more understanding in the general public as well, but also with breeders, with seed companies. The industry is often not, certainly for oil palm, it's often not vertically integrated in the sense, especially smallholder farmers, they're only responsible for growing the crop. They're not responsible for making the seed or even for extracting the oil. And so, you know, they only get a piece of the value chain. And it's therefore difficult, even if they're going to end up being the ones who benefit the most, it's difficult to, you know, persuade an entrenched, you know, a system that works well to change their ways in order to benefit these the people who matter most. But that is the challenge.
Great, thank you. So at this point, I think we will close out this portion of the program. Sarah, Rob, Ravi, and Mark, I want to thank you all so much for being with us today, for some really great talks on your work and discussion on what really is a very critical and globally relevant topic.